Okay, let's go ahead and get going. So, excuse me, we are touring the GI tract, and we are now into the small intestine, where we've got three accessory structures that are going to be important in the process here. So we've talked about the liver, which produces and secretes bile, the gallbladder, which stores and concentrates bile and then dumps bile into the small intestine as the chyme enters. The bile then, the primarily digestive function of the bile is to emulsify lipid, as we have seen in the class this week. So now we're going to deal with the pancreas, which has a major function in the chemical digestion processes that take place in the small intestine. So as the slide says, the pancreas secretes a pancreatic juice, which has two components into the small intestine. One of the components is bicarbonate. So it secretes a, relatively speaking, large number of bicarbonate ions, and the primary purpose of bicarbonate is to raise the pH of the chyme as it enters the small intestine. So typically, what is pH of the chyme? What is the pH of the chyme in the stomach? 2. And as it enters the small intestine, we need to raise the pH to about 5.5 to 6, 6.5 as the chyme moves through the small intestine. So that is done, of course, with bicarbonate ions. And I suspect all of us by this point understand how dumping bicarbonate ions into something will raise the pH. Excuse me again. And then the pancreas also will secrete a number of digestive enzymes that will be important in chemical digestion process. So what is the stimulus for this? As the chyme enters the small intestine from the stomach, that will stimulate the enteroendocrine cells in the small intestine, in the wall of the small intestine, to secrete cholecystokinin and secretin. And cholecystokinin then will stimulate pancreatic enzyme secretions into the pancreatic juice, and secretin will stimulate bicarbonate ion secretion. So we've seen cholecystokinin and secretin in a number of cases here, regulatory processes in GI function. And so this slide is just simply a summary of the action of these two enzymes. So you can take a look at that and put them all together, or put all of their actions together as shown on this slide. Okay, now, enzymes that are secreted in the pancreatic juice. So this slide we need to take a look at, and well, let's take a look at it. So we've got a number of, this left-hand column is a list of pancreatic enzymes in their active form as they appear in the small intestine. Okay? So a number of these enzymes, however, are secreted from the pancreas in the zymogen form. What's the zymogen form? Inactive. Okay? So these protein digesting enzymes, trypsin, chymotrypsin, elastase, and carboxypeptidase. Excuse me, I'm going to be coughing a lot, so hopefully I'm not driving your ears crazy. God, I love that. So we need to secrete these things along with this phospholipase, because phospholipase, of course, can damage phospholipids, which obviously are important in the wall of the lumen, or I'm sorry, in the wall of the duct leading into the lumen. So these 5 enzymes need to be secreted in this zymogen or inactive form. Then they need to be activated once they get into the lumen of the small intestine. So just to make sure we have the definition correctly, if anybody ever asks you, and I'm not sure who would, but if you're walking through the mall and somebody comes up and asks you for a list of enzymes, for a list of what is secreted in the pancreatic juice in the way of enzymes, this is the list. Okay. Okay, so these zymogens are what are actually secreted. And then these enzymes are fine to be secreted in the active form, so they are, because they won't damage the duct. So now, once these zymogens get into the lumen of the small intestine, they need to be activated. And the way this works is trypsinogen is activated by an enzyme that's, this is called, actually, when we'll look at this in a bit, a brush border enzyme. In other words, it is in the small intestine attached to the brush border, enterokinase. So enterokinase will convert trypsinogen to trypsin, which is the active form, and then trypsin, one of the actions of trypsin is to activate these 4 zymogens. Okay? So in other words, trypsin converts chymotrypsinogen to chymotrypsin. It converts proelastase to elastase, procarboxypeptidase, carboxypeptidase, and prophospholipase to phospholipase. And then the action of each of these enzymes is given over here. Okay? So this just becomes a learn what these enzymes are and what their actions are. And these actions are rather broad, so they should make sense. So in other words, we have protein digesting enzymes, we have lipid digesting enzymes, we have a carbohydrate digesting enzyme, and then some nucleic acid digesting enzymes. Okay, so we just mentioned the brush border. The border of the lumen of the small intestine, the endothelial cells, etc., contain villi and microvilli, which I'm sure you learned in anatomy. What's the purpose of having villi and microvilli? Why don't we just have a smooth border? Yeah, maximize surface area, because anytime, we've seen this multiple times, anytime you're trying to move something back and forth across membranes or any area from one to another, maximizing surface area is a very important thing. And so the surface area is much larger with the villi and then the microvilli associated with the villi. So if you look at the microvilli under a microscope, it has the appearance of a brush, which is why this is referred to as the brush border. Now, we then have enzymes or proteins that are attached to these microvilli. And those then, of course, are referred to collectively as the brush border enzymes. And here's a list of the brush border enzymes. We have 3 disaccharidases, sucrase, maltase, lactase. And I suspect with a limited thought input, you could describe what the substrate and products are of these 3 enzymes. Right? Okay. What would you imagine sucrase does? What's the substrate? Sucrose. Yeah, so it will hydrolyze sucrose. What will you end up with? What? Glucose and fructose. We all remember that from where? Yeah, Unit 1, if not before. Okay. Yeah, so maltase, lactase. So collectively, these things are referred to as disaccharidases. They hydrolyze disaccharide. Then we have a peptidase, aminopeptidase, and it probably, this name is actually fairly descriptive, fortunately. It cleaves the amino acid terminal from a polypeptide. I'm sorry, it cleaves the amino acid on the amino terminal of a polypeptide. So what would the products of that reaction be? Again, broadly, it would be a single amino acid and a polypeptide that's one amino acid shorter than it used to be. Okay. And enterokinase is a brush border enzyme, which we just looked at. Its primary function is to activate trypsinogen, which then will activate the other pancreatic juice zymogens. Phosphatases will cleave phosphate groups from nucleotides, and then we have a group of nucleosidases which cleave the base from the sugar. Okay. What's the purpose of all of this? Would you guess? Okay, so there's not enough motivation for guessing right now. So if I offered someone 5 bonus points for describing the purpose, would that motivate you? Not that I'm doing that. You guys already know I promise things and then never deliver. So, okay. So broadly, what are we trying to do here? We're trying to break down polypeptide or polymers, carbohydrate, nucleic acids, proteins, lipids, into monomers for what purpose? So that we can absorb them into the blood. Okay? Okay. So we also have some mechanical digestion processes, which in other words mixing the chyme, and movement. So we have to ultimately move the chyme through the small intestine. And the primary mechanical digestion and motility process in the small intestine is a process called segmentation. And segmentation looks kind of like this. And there are times somebody explains something, and even if it's obvious, the explanation sticks with you for a while. And so one of those explanations for me was this, segmentation. So this was back when I took physiology. I was 21, which means that was 44 years ago. And the physiology professor just said, so let's just imagine that we've taken out a small intestine and spread it out here. And we put chyme in it on one end, and we get about 8 students, and we all put our 2 hands around it. And then we just start squeezing randomly. What's going to happen to the chyme? Yeah, it's going to mix. Will it move? Sure. Yeah, I mean, if it's in here and I'm in and I'm on this end and I start squeezing randomly, it's going to move how? He then further gave the demonstration by saying, okay, I'm the chyme. Okay, so what? It's going back and forth and back and forth and back and forth as it's mixing, but ultimately it's moving down the small intestine towards the large intestine. But now, what's the difference between that and peristalsis in terms of rate? Peristaltic motion will move a food bolus through the esophagus much faster. But in the small intestine, we don't want it moving, the chyme moving rapidly, because that would make chemical digestion and absorption inefficient. And so we want a lot of mixing and slow movement, and segmentation accomplishes that for us. Okay, so now we're just going to take a look broadly at chemical digestion of these various biomolecules in the small intestine. So this will be kind of a broad overview. So carbohydrates, where in the GI tract does carbohydrate digestion begin? Yeah, in the mouth. Okay, so we have large carbohydrate polymers that salivary amylase will break up into smaller carbohydrate polymers, but they're still fairly large. And when it gets into the small intestine, then pancreatic amylase takes over and breaks down these starches mostly into disaccharides, but a few dextrins, which are just sort of 4 and 5 unit, 4 to 6 unit carbohydrate polymers. Then the 3 brush border enzymes, the disaccharidases, break the disaccharides down into monomers. And these disaccharidases will actually also work on dextrins and eventually then break all the carbohydrates down into monomers, which of course would be glucose, galactose, and fructose. Okay, proteins. We've got multiple protein digesting enzymes that collectively are referred to as peptidases. And we typically categorize these as endopeptidases and exopeptidases. And exopeptidases cleave a single amino acid off one terminal or the other of a polypeptide. Endopeptidases cleave polypeptides somewhere in the middle. Well, I shouldn't necessarily in the middle, but somewhere other than one of the terminals. Okay, so if you were going to name, or let me rephrase this, how many exopeptidases would there be then? Well, how many terminals do we have on a polypeptide? 2, right? What do we call them? What do we call them, Gabby? Yeah. No, I mean the terminals. The amino terminal and the carboxyl terminal, right? So what would you want to call the enzyme that cleaves the amino terminal amino acid off a polypeptide? Any guesses? Yeah, how about aminopeptidase? Good, okay, let's go with that. Okay, now, where do we find aminopeptidase? Where do we find enzymes here? Well, there's 2 locations, on the brush border, in the pancreatic juice. So aminopeptidase is a brush border enzyme. It's an exopeptidase that cleaves the amino terminal amino acid from the polypeptide. What do we want to call the other exopeptidase? Come on, be brave. Somebody yell it. Yeah, what would you want to call a peptidase that cleaves a carboxyl terminal amino acid from a polypeptide? Carboxypeptidase. Good one, yeah. Okay, so that is secreted in its zymogen form in the pancreatic juice, and then once it gets into the lumen of the small intestine, it's activated by trypsin. Okay, so then these other enzymes, trypsin, chymotrypsin, and elastase, are exopeptidases that cleave break peptide bonds somewhere other than just the terminal, okay, at the terminal amino acid. Now, we also, let's take a look at, there's another group of enzymes, or family, of dipeptidases and tripeptidases that that cleave, of course, dipeptides or tripeptides. These enzymes are actually located inside the epithelial cell. And it turns out that, that the apical membrane can transport individual amino acids, dipeptides, and tripeptides. The basolateral membrane can only transport amino acids. So we have to further hydrolyze any of the dipeptides and tripeptides that get into this epithelial cell by these two enzymes, or these two enzyme families. Okay. Lipids. Okay, so we know from the lab that lipids are emulsified by bile salts in the small intestine, and then pancreatic lipase, which is secreted in its in the pancreatic juice, hydrolyzes triglycerides into fatty acids and monoglycerides. And so this just kind of shows this pattern. So bile salts from things will emulsify fat droplets, and then lipase will hydrolyze these triglycerides into free fatty acids or monoglycerides, which are then packaged into molecules referred to as micelles. And then nucleic acids. So nucleic acids are hydrolyzed by ribonuclease and deoxyribonuclease, which are pancreatic juice enzymes. And that will then break down nucleic acids into nucleotides. So then nucleotides are further hydrolyzed into their three component parts. So the three component parts of a nucleotide are phosphate group, sugar, and nitrogenous base. So these pentoses, phosphates, and the nucleosidases and phosphatases, which are brush border enzymes, break down the nucleotides into their component parts. Now, absorption. So absorption is, and again, those of you that go on and take advanced physiology classes usually means you spend a whole semester in one system. Like you might take a GI physiology class, which means 15 weeks of GI, which typically means about 3 or 4 weeks of all these absorption mechanisms. Okay? How do you absorb a phosphate group across the apical membrane and the basolateral membrane? How do you absorb glucose monomers? How do you absorb everything? And go through the details and worry about how gradients are established, how electrochemical gradients are established, and how they are influenced by things. Okay, we're going to cover this in about 90 seconds. Yeah, maybe about 4 minutes. Okay. So we need to just kind of look at this broadly. And glucose and galactose cross the apical membrane by secondary active transport. Now let's just use this as a bit of an opportunity to review, which is always fun. What is secondary active transport? Yeah, so secondary active transport, I mean, it's active transport, so something is being moved against its electrochemical gradient, which means there has to be some kind of energy source. And the energy source for secondary active transport is typically some ion moving down its gradient, which releases energy to drive the transporter, which is either a symporter or an antiporter, across. So what we're going to do here is anything that is moved across secondary active transport by sodium is going to be symport. And the majority of sodium transport is symport. Anything that goes across by hydrogen, we're going to call it antiport. secondary active transport with hydrogen. Okay? So the terminology you should use here is glucose and galactose cross the apical membrane by sodium symport. Now, when you say that, of course, you understand that that means that it is secondary active transport driven by sodium moving down its electrochemical gradient. Fructose crosses the apical membrane by facilitated diffusion, and all three of the monosaccharides cross the basolateral membrane by facilitated diffusion. Amino acids, individual amino acids cross the apical membrane by secondary sodium symport. Dipeptides and tripeptides cross the apical membrane by hydrogen antiport, where they are further hydrolyzed in the epithelial cell by the dipeptidases and tripeptidases. And then all of them cross the basolateral membrane by, add a word here, facilitated diffusion. Lipids, all the lipids cross both membranes by simple diffusion through the phospholipid bilayer. Okay, now, lipids, these chylomicrons, which, I guess I should put that, my cells are broken down at the membrane to long-chain fatty acids and monoglycerides, and those are what cross the apical membrane. Then once they get in here, they are packaged in a different type of structure referred to as a chylomicron. The chylomicrons then are absorbed into the lymph and move through the lymph system and then are returned to the blood at the port here in the left subclavian vein. Everything else is absorbed into the blood directly. Okay. I'm going to today kind of just run through the first 10 or 12 slides or so, which are mostly nephron anatomy. And I'm just going to kind of mention this really broadly. But the first thing we want to do is, what is the function of the kidney, the renal system? It is, in short order, to regulate the composition, volume, and pressure of the blood. Okay. So that's the simple way to say it. That's hugely important to maintaining homeostasis. Regulate the composition, volume, and pressure of the blood. So what does that all entail? Well, you know, plasma ionic composition, plasma osmolarity, plasma volume, and volume goes a long way to regulating pressure. Plasma pH, remove metabolic waste products, etc. Okay. So what does this have? Well, we've got 2 kidneys. Each of them have a ureter running from them to the urinary bladder, and then a urethra which runs from the urinary bladder out of the body. If we look at the macroscopic anatomy, we have a number of structures here that you studied in anatomy, and I'm not going to go over them in a major way. The macular structures, you can just kind of review those and just be able to, if you don't remember what these things are, just kind of look at them and identify them on this aspect. But the thing I want to spend our time with mostly is the nephron, which again is what we did in the lab this week. So I suspect most of you are familiar with it. So So we have the renal corpuscle that filters the blood. So that consists of Bowman's capsule, which is this, I'm not sure what, just looks sort of like this. And the glomerulus, which is the inner portion of this, which contains the glomerular capillaries. And then we have afferent arterioles bringing blood into the capsule, efferent arterioles moving blood away from the capsule. Then we have the renal tubule, which extends from the capsule through the proximal convoluted tubule, loop of Henle, distal convoluted tubule, and collecting duct. And then we have multiple peritubular capillaries, which are associated with the renal tubule. We can broadly look at nephrons in two categories, depending upon how far they dip into the medulla. So the ones that are mostly in the cortex are referred to as cortical nephrons. The majority of nephrons are cortical, but then we have some juxtamedullary nephrons where the sort of the fine-tuning of the reabsorption and secretion takes place. And then we have this juxtaglomerular apparatus, which is located sort of a group of cells located here at the intersection, if you will, of the afferent arteriole, efferent arteriole, and the distal tubule. And there are a number of secreting cells that are located in this juxtaglomerular apparatus, one of the primary ones being renin, which we've seen in the aldosterone pathway, angiotensin II pathway, which plays a big role in regulating blood pressure, which is, of course, important in all of our activities. Kidney blood supply. Again, we've got a renal artery that moves into an arcuate artery that eventually leads into the afferent arteriole, glomerular capillaries, efferent arteriole, peritubular capillaries, and then blood leaving. Okay. We will pick up from here on Monday. We'll start talking about renal function Monday.